Okay, now it's quiz study time, guys. So I'm going to be going over some of the main things to know, we need to know for the quiz. So first, a bit of a recap on the last quiz. So the first question was about the three functions of the kidney. So filtration, reabsorption, and secretion. The second question was on the afferent and efferent arterioles. So the two things we need to know, those two major vessels. Allow blood in and out of blood. Or we could have said it mesangial cells. Afferent arterioles dilate, efferent arterioles constrict. Lots of reabsorb. And then third question about reabsorption of water or amino acid, fatty acids, solutes, glucose, organic things, ions being removed from the filtrate, placed back into the interstitial / peritubular spaces, which will become part of the capillary flow. Sending back solutes and the water is passively flowing. Using this figure. Based on this figure of the nephrons. Lots of water going into the proximal convoluted, but some are left in the tubules. So if you have decreasing blood pressure, your afferent arterioles dilate. If you have increasing blood pressure, your afferent arterioles constrict. The kidneys need to function with steady amount of pressure at the glomerular filtration site. Okay, next figure. Next figure is this figure of the proximal convoluted tubule. We're going back to the PCT again. So we don't need to memorize all of the exchanges going on. There's a lot of things already filtered out of the blood through filtration coming out as a capsule in the filtration process. Okay, so there's a table here I wanted to show you guys of the ions. So here's a bit of the table. I was talking about with the types of ions, what is being secreted and reabsorbed and what is not. So what is reabsorbed is ions and metabolites. Oh no, wrong figure. So the table. So a lot of things are already being filtered out of the blood through filtration, coming out as a capsule in the filtration process. As you saw in that figure, we have ions and metabolites being reabsorbed. We have ions, waste, and other misc are being secreted. And some have no transport mechanism, such as urea, water reabiligens, and bilirubin. Okay, on to this table. Continue on with the table. So there's tubular reabsorption and secretion going on. Reabsorbed ions and metabolites. So there's reabsorbed ions and metabolites. Secreted ions, water, waste, and misc. No transport mechanism needed for some. Going into surrounding spaces and eventually goes back into the blood. Diffusion, osmosis, and active transport, most of these follow these processes. We don't want to deplete our blood of water, so water is constantly following these solutes. Taking water out of urine and leaving blood at low volume, low pressure, and low flow. Blood is always trying to go where water goes. Next figure, or back to the figure of the PCT. The nephron loop, or the loop of Henle, just open that. Nephron loop, or loop of Henle, is where sodium ions, chloride ions, and water are put in their places. The loop is where the kidney has the chance to hold water in urine and get water back in the blood. Okay. Urine is the place where blood gets to reclaim water. It's where the kidney gets a chance to hold onto solutes, dump it in urine, and get water back from the blood. Next figure is this, so you can see the nephron loop. A bit more of a detailed picture of it here, and the types of things that are able to escape and some things that are not. The ascending loop has mostly active transport of sodium and chloride ions, where urine is concentrated and rehydrated. controlled system where sodium and chloride are blocked, it's impermeable to solutes. So sodium and chloride, those are the solutes. So if more solutes are going out than water, then you lose more water. So more concentrated solutes are cannot enter by diffusion. So if more water leaves, the loop is contributing to the outflow of water. Filling spaces so that water is allowed to leave. So it's also impermeable to water on the way up. So functions of the nephron loop: concentrating urine. The nephron loop is responsible for concentrating urine, reabsorbing water into the blood. The loop is controlling the outflow of blood and outflow of water and solutes on both sides. The main mechanism for ensuring water gets back into the blood, whereas solutes don't. What a professor asked the class, so what is the function of the nephron loop? So the function of the nephron loop is for concentrating the urine, concentrating it by removing water and solutes, because the solutes go out, water is going to follow it. So it would be reabsorbing water into the blood. So that's the function of the nephron loop: concentrating urine, reabsorbing water. Water goes out of the if water goes out to the blood, sodium and chloride ions are available to the blood, and urea is also on its way out. Next figure, figure 2615, another view of the loops. You can see a bit of the hormones also in this process, a bit more. Yeah, ADH, antidiuretic hormone. Okay, so distal convoluted tubule. The collecting ducts collect from several different nephrons. The DCT is where there is variable secretion and absorption. The DCT and collecting duct responds to conditions in the blood, not in the urine. Blood conditions. Sometimes allowing more water in, sometimes not allowing water in. We don't need to worry about all the ions and how they are transported. Just sodium, chloride, and urea are the big ones. Next figure. Back to the loop figure. Or more on the DCT and the collecting duct portion. So the DCT and the collecting duct is impermeable to urea. DCT looks different from the PCT because the lumen is clear in the DCT, and the lumen is cloudy and fuzzy and pink in the PCT because the PCT has microvilli, which are actively absorbing and secreting tubular fluid. It's a more concentrated process in the PCT. In the PCT, one of the things being put back into the blood, leaving the tubular fluid heading out into the cells. Into the interstitial spaces and back into the back into the blood is glucose. Next figure, back to the PCT figure, showing water reabsorption and solute reabsorption. If you have high levels of glucose or hyperglycemia/diabetes in the blood, at the kidney, it will filter everything like a sieve. high glucose, you'll filter anything like a sieve, and then you put a lot of glucose in the PCT. Then the glucose is trying to get back into the blood at the PCT, but there is only so much length of PCT and time for for these ions and molecules to get out of the tubing and back in and back into the blood. So if you have high glucose levels, by the time you get past the PCT, you may have some glucose left in your urine and your filtrate, because if they didn't have enough time to get back into the blood. If you have glucose in your blood, something other than is a detectable issue. Glucose should not be showing up in urine. If glucose is in your urine, you should get checked. Question she asked the class is, what does a lot of protein in your urine mean? It means your liver is already broken down in blood, probably amino acids. Why not urine? Proteins are big. Too much glomerular pressure. Why not urine? Because proteins are too big, too much glomerular pressure ramming it through the sieve. So if we have lots of protein, it means your liver is not doing its job of breaking down those proteins before it reaches your urine. And oh, here's the quiz, guys. Here's the quiz. There's the grade. I attached it onto my notes. Yeah, a bit of a recap again. Glucose should not be in your urine. Glucose should not be in the blood, or else the liver will suffer. Hyperglycemia leads to diabetes type 2. So glucose is supposed to turn into glycogen for storage. Urine has some sodium and urea. I'll go over this quiz one more time. So the 3 functions: filtration, reabsorption, secretion for question 1. Question 2 was more on the autoregulation side. It's done by afferent and efferent arterioles dilating and constricting based on the pressure. Adjust to the amount of adjust its mouth to allow blood to enter. If you have decreasing blood pressure, your afferent arterioles will dilate. If you have increasing blood pressure, your afferent arterioles will constrict, reducing the flow into the glomerulus. Sodium chloride and water should be most likely to be left in the tubular fluid and is less likely to be reabsorbed into the blood capillaries. Okay, figure 26-7. Back to that figure of the different types of nephrons. And then straight to 2613B. So DCT and collecting duct. After reabsorption, you got a lot of H2O, sodium, and chloride, urea, and stuff left in the filtrate and tubing. Now the kidney has the problem of regulating the sodium and chloride ions in the blood. Regulating the amount in them, the movement placed back into the blood is how much left in the urine. Liver given urea picked up through the filtration and put into the tubules. Urea levels, how much nitrogen you've been eating, how much protein you've been eating, and how much has your liver formed by combining ammonia with carbon dioxide. That's urea, or how much urea you have. Sodium, chloride, and water are adjusted at the nephron loop. Descending limb is, in that figure, the red line is shows that it's impermeable to solutes. Mostly water is allowed to leave, moving to higher fluid concentrations in the tubular fluid. For the descending limb. For the descending limb, there's a high osmotic concentration of 300 millosmoles per liter in the beginning. and then that increases all the way to 1,200 milliosmoles per liter. Because water is leaving, so there's more solute concentration in the DCT. So how are we getting back to a lower concentration? We get solutes out such as sodium and chloride. Ooh. So the liver gets urea, ammonia plus carbon dioxide, picked up through filtration through the kidney tubules, works its way to the urine for excretion. A little goes back into the blood for osmotic effect on the blood. Okay, ascending limb. So the black line shows that it's impermeable to water barrier. We can't put any more H2O back into the blood, but sodium and chloride get regulated by active transport. By the end, concentration is similar to what we started, but we lost a lot of water, so H2O, sodium and chloride back to the blood, so volume is much lower. Impermeable to water, going from 1,200 milliosmoles per liter all the way down to 800, then finally down to 100 milliosmoles per liter. Very close to the starting amount of solutes we had in the DCT. In the descending limb. About the same concentration but at a lower volume. So PCT to DCT, DCT comes through the back of cells of the juxtaglomerular capsule, or JGC, up ascending loop to the DCT and then to the collecting duct. Next figure. So, into the cortical nephron. And then next figure again. Some reabsorption into these, so capillaries are in a bundle. Some reabsorption in the DCT and PCT and beginning of secretion. And beginning of secretion. Secretion is an active form of moving things across the membrane, so from blood to the filtrate. So reabsorption is filtrate or tubular fluid to blood, and secretion is blood to tubular fluid. It's the blood's last chance to secrete stuff into the urine that doesn't want to have active transfer from blood to filtrate. So what is being reabsorbed? Some urea is going back into the blood, as well as some sodium and chloride and water. So what is being secreted from blood into the DCT? And ADH, antidiuretic hormone. Okay. Secrete from blood to DCT, variably hydrogen ions and bicarbonate ions. The kidney can take on more the hydrogen and bicarbonate ions. It's the blood's last pass at regulation of the kidney pH. If blood pH is too low, it might secrete more hydrogen ions into urine filtrate to hold on to bicarbonate ions. If blood pH is too high, 7.45, it's going to hold on to its hydrogen ions. And secrete its bicarbonate ions. So variably would be variable reabsorption, hydrogen, and bicarbonate ions. Are reabsorbed to DCT. From blood. So secreting is like removing from the blood. pH is something blood is sensitive to, such as lungs and kidneys. We have chemoreceptors that alert pH levels. Phosphate, PO4 3-, and potassium can also be secreted. Metabolites are also secreted from cell metabolism, giving off various ions as byproducts, such as carbonic acid, CO2, bicarbonate, and ketones. Ammonia is created through fermentation, must be detoxified. Alcohol is created through fermentation. Cells produce these as byproducts that get detoxified in the liver, dumped into the blood. The kidney extracts ammonia and then excretes them. So metabolite levels, carbonic acid, bicarbonate, ketones, and ammonia levels must not be too high. Where do you get phosphate and potassium from? Bananas, vegetables, diet, and kidney is a dumping ground for high levels of anything. Excretion versus secretion. Excretion is leaving the body through pores in your skin, urinary system, digestive system, exhaling of carbon dioxide. Secretion is active transport across the membrane. So from blood to the filtrate, basically. Excretion is out of the body. Secretion is active movement of its molecules from the blood and back into the filtrate. Blood to filtrate. Secretion, blood to filtrate. Or back into the tubules. Good and bad drugs. Drugs and metabolites like opioids, once they do their job, the body gets rid of them. Too large to be passively exchanged across the membrane must be actively secreted from blood to urine. Rate will vary according to the needs of blood. Chemoreceptors will vary. At the DCT, there is variable secretion and reabsorption, not a guaranteed rate based on needs of blood at the current moment. So chemoreceptors and baroreceptors have an effect on what is happening. Next figure, the spotlight. So next figure is a very complex one. This one takes up 2 pages. Let's jump into it. She wants us to read all 8 steps. Collecting duct is not a technical part of the nephron because one collecting duct is serving several nephrons. The collecting duct is the blood's last chance to alter sodium, chloride, and water content in the blood. It alters urea content in the blood. Likes to keep a little urea in the blood because it is a great osmoregulator. As you get rid of sodium and chloride in blood, some of the urea is kept in the blood. At the nephron loop as well, because there has to be some way for blood to keep pulling water by osmosis. Urea is a great osmopuller. At the collecting duct, there is some push of urea back out of the duct and into blood. Water is still leaving the collecting duct and heading back to feed the blood. Table 26.1. Cells that make up the nephron summary. So charts for reviewing cells that make up the glomerular capsule and surrounding the glomerular capillaries. So the PCT has microvilli at the edge, making it seem a bit fuzzy. Nephron loop has skinny part is stratified squamous, thick part is simple cuboidal. DCT is mostly cuboidal without microvilli. The papillary duct has the columnar cells that extended overlap. The collecting duct down into the lower part of carbon monoxide turns into columnar lining cells. Papilla at the base of CO. At the base of collecting duct. Papilla down into the lower part of the collecting duct, it turns into columnar lining cells. The papilla at the base of the collecting duct of pyramids in cell work. The juxtaglomerular complex has receptor cells that receive sodium ion levels. In order to regulate, you have to be able to perceive current conditions using receptors, blood receptors, pH, pressure. Negative feedback loop, adjusting current conditions so that the effects alter current conditions to a set point. 2611. Oh no. So first step, autoregulation regulates pressure and afferent at the filtration slits. Too much pressure wrecks the filtration net. The pressure in the glomerulus must be just right. Afferent and efferent does this by regulating pressure in the glomerulus. If you have low pressure, the afferent arterioles will dilate to allow more fluid in through little packets of capillaries. High pressure afferent arterioles will constrict, reduce the amount of blood in glomerular capillaries, reducing the amount of blood done in order to increase blood pressure in the body. If you're trying to increase blood pressure, so there's the local picture, autoregulation. If you're trying to increase blood pressure, you dilate the afferent and you constrict the efferent. If you're trying to decrease blood pressure in the glomerulus, you constrict the afferent and you don't need to do anything to the efferent. If the afferent arterioles are has too much blood going to the glomerulus, it will slow down that flow by constricting the efferent. Does not alter if the efferent arteriole sense low blood pressure will dilate and efferent will constrict, filtering more blood in local picture. Pressure sensed locally by JG cells on afferent arteriole buoy. So pressure is monitored by juxtaglomerular complex cells, receptor cells, and based on that, the afferent arterioles and efferent arterioles will dilate or constrict based on the pressure senses by JG cells. If you have low blood pressure, the filtration site, you will not be able to filter much fluid through. Low blood pressure, low fluid filtration. The JG complex detects sodium levels not in blood but special cells in the DCT. DCT has what's left through the nephron loop. So what's left, these cells sense how much sodium in the filtrate at this point. Sodium levels reflect potassium levels because the two are usually in opposition. High sodium on side of membrane means high potassium on the other side. If pressure is low in the glomerulus and not filtering much fluid away, not much sodium in the DCT because there was not enough pressure pushing it out. Low blood pressure at the glomerulus means low sodium, low liquid, low chloride, and at the DCT. Second way of determining blood pressure at the kidney: blood pressure at the kidney is not where it should be, then the kidney cannot do its job. It can only do its job if there isn't not enough. It cannot do its job if there's not enough push at the glomerulus to do its job. Okay, we have to end here. Next is all about hormones, hormone properties. I gotta go take the anatomy quiz. Thanks guys for sticking around for this video. If you watched the whole thing, watched all the studying, got something to go crazy. Let's see how this quiz goes.